In this video, we present a novel method to automatically find the heart, lungs and liver of the fetus in magnetic resonance images. The main challenge when imaging the developing fetus is fetal movement. Additionally, when searching for the fetal organs, we are faced with the arbitrary orientation of the fetus, as well as the variability due to fetal development. In order to accommodate for fetal motion, the images are acquired as stacks of 2D slices that freeze in plane motion, but may form an incoherent 3D volume. Motion correction can be performed retrospectively to obtain a high-resolution volume from several stacks of 2D slices. The automated localization of fetal organs can be used to initialize this motion correction procedure. In order to reduce the variability due to fetal development, we propose a size normalization based on the gestational age. To account for the arbitrary orientation of the fetus, we use image features steered by the anatomy of the fetus and propose to detect fetal organs sequentially. Starting from the brain, we find possible locations for the heart, then for the lungs and liver, before selecting the most likely position of each organ. To reduce the variability due to fetal development, we resize the images based on the gestational age of the fetus. This allows to train a single model across all gestational ages. This size normalization uses the crown rump length estimated from the gestational age. As we perform a sequential search, starting from the fetal brain, we know that the heart is within two spheres of radii R1 and R2. For each possible location of the heart, the lungs and liver must be within the sphere of radius R3, centered on the heart. R1, R2 and R3 are independent of the gestational age, thanks to the size normalization. At training time, 3D features are extracted in a coordinate system aligned with the fetal anatomy. At test time, 3D features are extracted in a rotated coordinate system. The brain fixes a point, while the heart fixes an axis. In an analogy with the half forest, we perform first a classification step, followed by a regression step for the heart, then for the lungs and liver, classification step, followed by a regression step. We score the different combination of organs candidates based on the regression votes as well as the relative position of the different organs. We now present some localization results. The acquired images are shown on the left, and a denoised version can be seen on the right. Each cloud point corresponds to a different organ, in blue for the heart, red for the left lung, green for the right lung, and yellow for the liver. Those point clouds correspond to the back projection of the regression votes. The effect of denoising can mostly be seen in cases of sudden fetal motion. We propose the size normalization, a sequential localization of fetal organs, and features steered by the anatomy of the fetus in order to localize the heart, lungs, and liver of the fetus. Our approach combines classification and regression forests. We train a single detector across all gestational ages, and in 90% of the tested cases, the detected heart is within 10 mm of the ground truth. For more information on the source code, please visit our website.